Well, first, I'd like to thank the organizers of CannMed, and thank you so much for the gracious invitation to present this year. And I'm so sorry and so disappointed I can't be with you in person. I'd much rather be there, trust me, perhaps next year. In the meantime, I'm honored to share some data with you from basically a first look, if you will, at open label data from a CBD clinical trial for anxiety, or what I like to call high anxiety, question mark. When we think about anxiety disorders, it's important to remember that they are the most common of all mental health conditions. According to the World Health Organization, 1 in 13 globally suffers from anxiety. Lifetime prevalence of anxiety disorders does vary across age groups. There are different examples. For instance, within the United States, it appears that between the ages of 30 and 44, we have the greatest likelihood of having an anxiety disorder. Interestingly, and perhaps not surprisingly, the COVID-19 pandemic has not helped the situation. US Census data revealed 42% of people reported symptoms of anxiety or depression in December of 2020. That was an increase of 11% from the previous year. That's a pretty big jump, a 31% increase. So important to keep these things in mind. Anxiety disorders are generally underreported and undertreated, and lost productivity as a result of anxiety and depression costs the global economy somewhere in the neighborhood of about $1 trillion every year. So it's not as if there aren't treatments for anxiety disorders. There are plenty of available pharmacologic approaches, including, but not limited to, things like SSRIs, SNRIs, benzodiazepines, MAOIs, tricyclics, 5-HT1A receptor agonists, anticonvulsants, atypical antipsychotics. That's not the issue. The problem is that despite a number of options with regard to conventional medications, response rates remain somewhat limited, and residual symptoms are often present. That's a problem. In addition, adverse effects also limit tolerability and adherence. I don't know how many times we've heard people say, yeah, I took the medication, but the side effects were so hideous, I had to stop. So limited efficacy of current pharmaceuticals and people's unwillingness to continue with something that causes side effects underscores the need for alternative treatments for those suffering with anxiety. The growing availability and popularity of legal cannabis products, combined with higher rates of anxiety globally, further exacerbated, of course, by the COVID-19 pandemic, has reignited a long-standing conversation about the potential effects of cannabis on anxiety. It's important to remember, though, that when we think about cannabis, we again, as I'm very fond of reminding people, we have one word, and we use it to describe lots of things, almost anything at all from the plant. When we think about products, cannabis or cannabinoid-based products, very important to remember that they can either be plant-derived or non-plant derived. So in terms of plant-derived products, you can have full-spectrum products, which contain a host of cannabinoid constituents, delta-9 THC, CBD, a host of other cannabinoids, as well as terpenoids, the essential oils that give cannabis its characteristic scent and flavor profile, and have their own biobehavioral effects, flavonoids, etc., you name it. Then there are broad-spectrum products, which contain all of the above except for delta-9 THC. Interesting and a completely separate talk, probably, in terms of how we, quote, get to zero, because it's an interesting approach. But broad-spectrum products are designed to capitalize on the full spectrum aspects of having everything from the plant that may be beneficial without the potential intoxicating issues related to THC. You can also have plant-derived isolates, for example, things like Epidiolex, a single extracted compound, cannabidiol, from the plant that's purified, so it has no other cannabinoid constituents, no terpenoids, no flavonoids, etc. Isolates can also be non-plant derived, so things that are more conventional medications, for example, what we would call a synthetic cannabinoid like nabilon. So it's not just one thing. Cannabis and cannabinoids can be used for multiple reasons in many ways and in countless combinations, so it's important to keep that in mind. Single compounds, like the single isolated cannabinoid or compound that I talked about, basically works very differently from a full spectrum product, right? Where you have the broad range of naturally occurring chemicals in cannabis, including THC, and the entourage effect, or the synergistic action that occurs in the presence of multiple cannabinoids, terpenoids, and flavonoids, is something that can be leveraged. There are many who question the presence of things like the entourage effect. I think it's becoming more and more widely accepted at this point, especially since lower doses of full spectrum products may in fact have therapeutic effects relative to single extracted compounds. There was a meta-analysis in epilepsy patients conducted in 2018 that basically looked at patients using CBD-rich products. These were full-spectrum products, and the meta-analysis basically reported that lower doses were required to achieve a therapeutic effect, and fewer adverse events were reported compared to those patients who were taking purified products or single extracted compound products. So important to keep in mind. The relationship between cannabis and anxiety is also a complicated one. There's been research suggesting that cannabis use is prevalent among individuals with anxiety disorders. And so the question becomes, what are we really looking at? Relaxation and relief from anxiety are among the most commonly reported motives for cannabis use. And to date, very few studies have examined the causal nature of the association. And those that have failed predominantly to control for what I would call related factors, which has led to mixed findings. Most at this point believe that the etiology of anxiety disorders is multifactorial and not likely caused by a single factor, including cannabis use. So that's very, very important to keep in mind, but there are still plenty of people who are convinced that that is probably the cause and not a solution. There have been several reviews that have looked at the effects of cannabis on anxiety. Going back to 2012, there was a review that looked and basically reported that outcomes were highly variable. Cannabis may have a bidirectional influence on anxiety responses as a function of dosage. Not incomprehensible, certainly. A later study noted mixed findings with regard to impact of cannabis on anxiety in recreational consumers. And I should say right here, it's a very, very big, sort of a big part of the picture, whether we're talking about individuals with anxiety disorders who are being treated as those using recreationally and those using for, quote, medical purposes with regard to how they're considered and how the approaches may vary. In a systematic review in 2017, there was a report that concluded that cannabis use is no more than a minor risk factor for the development of elevated anxiety symptoms in the general population. And in fact, the National Academy of Science, Engineering, and Medicine report in the same year reported there was limited evidence that cannabis or cannabinoids would be effective for improving symptoms of anxiety and PTSD. So why so mixed? Individual factors, including but not limited to things like your genetic profile, physiology, other substance use, and previous experience with cannabis, are very likely to play a role. The impact of cannabis and cannabinoid products on anxiety appears to be related to the very specific and very different impact and effect of individual cannabinoids. For example, research findings suggest that delta-9 THC and cannabidiol have different effects on anxiety, and that dose is absolutely a critical consideration. When we think about THC, we know that THC can be anxiolytic at low doses, that is, it can reduce symptoms of anxiety, but is often anxiogenic at higher doses, so it can either exacerbate or perhaps even cause anxiety at higher doses. There have been a number of studies demonstrating this with regard to acute administration. I think the earliest one that I have on this slide goes back to 2012, where healthy males administered 10 mg of THC exhibited increased anxiety as well as dysphoria and some psychotic symptoms, sedation and subjective intoxication, as well as increased heart rate. Some later studies found that daily use was associated with anxiety disorders even after stopping the use of cannabis. Acute subjective effects following smoking joints that contained up to 60 milligrams of THC in recreational consumers demonstrated that THC decreased calmness, alertness, and contentment, and THC-induced increases in anxiety actually lasted up to 8 hours after smoking. Among cannabis users, female users report increased anxiety symptoms relative to female healthy controls. There may be some obvious gender differences. On the other side, CBD, it appears, perhaps, has anxiolytic properties. There have been a number of studies going back into the 80s, actually. Our folks outside of the US have done some really compelling work. The very first one looked at THC-induced anxiety. CBD was able to mitigate the anxiogenic effects produced by THC. The same group actually also reported that CBD reduced anxiety induced by the simulated public speaking test in healthy controls and in patients with social anxiety disorder. And there was a more recent clinical trial, a double-blind clinical trial in Japan that looked at the efficacy of 300 milligrams of hemp-derived CBD isolate, so single extracted compound, in teenagers with social anxiety disorders and avoidant personality disorders. The CBD treated group exhibited significantly decreased anxiety relative to the non-treated group. So again, rather compelling. With regard to dose, investigations of all use study products containing single extracted compounds, CBD only, purified CBD, which exhibits an inverted bell-shaped dose response curve with limited dosage range for therapeutic response. That's important. Remember that whole plant full spectrum products have been shown to yield therapeutic response potentially at lower doses relative to single compounds, likely the result of the entourage effect or this synergistic action of the cannabinoids, terpenoids, and the other players. So really, really interesting. Just keep that in mind. Everything that's been done so far has been using single extracted compounds. So in 2014, I started a program called MIND, or Marijuana Investigations for Neuroscientific Discovery, and we have a number of observational, longitudinal, cross-sectional survey and clinical trials underway. The very first clinical trial we launched some years ago is a cannabis-derived high CBD formulation for individuals with moderate to severe anxiety. We have a number of other trials ongoing for anxiety as well, which I'll tell you a little bit about, but this is the one I'm going to show you some data from. This is basically a custom-formulated study product that I sourced. The original story is actually much more interesting than the, than the final story, but the study product wound up being formulated with a NIDA-based extract. We've completed the open label phase of the open label to double blind. Double blind is now underway, and we actually have added a third arm. So it was an open label phase. The double blind phase includes placebo, the same product that people got in the open label phase, and a single extracted isolated version of the study product. So study product removed everything except the CBD. That's underway. And I'm going to show you data from the open label phase in just a minute. We also have a CBD-related project focused on patients with anxiety and agitation. Basically, I formulated that product with some industry partners. The open label phase of that study is underway. I also have a hemp-derived high CBD formulation for folks with anxiety who don't quite need the same level of severity for the very first trial. Again, formulated in partnership with some industry collaborators, and that open label phase is underway. I just got another IND approved for patients with a mood disorder. So in terms of the anxiety realm, we have a lot going on. So in terms of clinical trial product formulation, the products are custom formulated by myself and my program in collaboration with the NIDA Drug Supply Program, as well as some industry partners and chemists with what I would call significant cannabis experience. It's basically how you go from taking an extract that looks like that, which is this very, very gummy, rather unfortunate looking and smelling product that patients are definitely not going to be able to use turnkey, and turning it into something that looks like that, what you see in the dropper. It doesn't taste too bad, actually, I have to say. So this very first ever clinical trial of a whole plant-derived high CBD full spectrum product for anxiety is, like I said, open label to double blind, custom formulated solution, 4 weeks of active treatment, and basically the study product consists of approximately 10 mg per mil of CBD, which doesn't sound like a crazy high dose. It's administered 3 times a day. It also has somewhere in the neighborhood of 0.24 to 2.6 or so mgs of THC. So at a 3-time-a-day dosing, you're under just 1 milligram of THC per day. Under 1 milligram of THC per day. Keep that in mind. It's sublingual administration, and patients are taught how to do it in the lab, and then they do it on their own at home. This is a quick overview of the study timeline. Basically, we have a screening visit where everyone gets a full clinical and diagnostic assessment. They complete a series of anxiety and mood rating scales, sleep, quality of life, and other scales, a full neuropsych assessment, obviously urinalysis, and for some of our folks early on, we did a blood draw for PK/PD work. We then assess them every week with regard to anxiety and mood scales, as well as clinical check-ins and side effect assessments and urine screens. The pandemic shifted this at the very end of the trial, I will tell you. Neuropsych assessment is done again at the end of the 4-week clinical trial, and basically, at the end of the 5 weeks, we're able to verify how much they took and sort of back calculate. Every week that they come in, or every other week, sorry, in the open label phase, they came in every week. We actually weigh their bottles on the way out and on the way in. So it's not just us telling them take 1 mil 3 times a day and then reporting, having them report what time they took it, etc. We actually weigh the bottles out and weigh the bottles in. We assess their clinical ratings and their cognitive performance, and we're, of course, interested in the safety profile and potential for serious adverse events. Urinalysis was completed to look for THC positive status over the course of treatment, which is another really important and rather interesting finding from this study. So what do we do in terms of anxiety measures? The Beck Anxiety Inventory is a self-report measure. It's a 21-item tool that can be used to screen for anxiety, and each item is rated on a scale of 0 to 3. It's a descriptive of subjective, somatic, or panic-related symptoms of anxiety. Then there's the OASIS, which is also self-report. It's only 5 measures that can be used across anxiety disorders to measure what we call functional impairment caused by anxiety, and it measures both frequency and intensity of anxiety, as well as the degree of avoidance and interference with work and social function. Finally, there's the HAM-A, or Hamilton Anxiety Scale. It's a clinician-administered scale, a 14-item metric that is basically administered in the form of an interview. This allows for information for multiple sources to influence ratings. So we have the subjective report, we have the examiner's observation, and it's been shown to be one of the most, if not the most, reliable index of clinical state with regard to anxiety. So in terms of this particular cohort, this was a group of 15 folks. We set out to complete open label data, or collect open label data from 15 folks. We stopped at 14. Actually, we set out to collect data on 16 people. We recruited 15 and 1 was discontinued, which I'll tell you about in 1 second. Average age was about 41, 16 years of education. And as you can see from their scores on the OASIS, the BAI, and the HAM-A, they all had moderate levels of anxiety overall. Of the 15 patients we enrolled, we discontinued 1 for the use of another cannabinoid product one time with their partner. That was enough for us. We had to let them go. Overall, the study drug was well tolerated, and there were no serious adverse events reported at all. Interestingly, and very importantly, no patients reported intoxication. On average, patients used not 3 mils per day, but 3.48 mils per day. The daily equivalent to that is about 34.7 milligrams of CBD and just about 0.8 mgs of THC. So that's why it's so important for us to weigh the bottles, not just take their word that they're taking about a mil. Again, they have a marked dropper, but still, people tend to administer slightly different levels over time. So what do we have? In terms of our open label results, when we look at the BAI, what we find is a 79.9% decrease in self-report ratings. So this is a significant improvement in ratings of anxiety. They go from about a 20.3 at baseline to a 4.07 at the end of the 4-week trial. On the OASIS, we see a 70.2% decrease. The scores go from 11.3 approximately to 3.3. And on the HAM-A, that's the examiner-driven scale, they go from just under 20, or 19.8, to 3.3, an 82.9% decrease. So these are rather striking decreases in self-reported anxiety. At some point, this will move forward. When we look at other ratings of mood, the Beck Depression Inventory, for example, a very commonly administered metric designed to measure self-reported measures or symptoms of depression. Our subjects go from a 20, approximately at baseline, to a 4.4. That's almost a 78% decrease in self-reported depressive symptoms. Profile of mood state, which allows us to look at differences over time. Total mood disturbance is sort of your grand total, goes from 65.8 down to a 23.9 between baseline and 4 weeks of treatment. That's almost a 64% decrease, so pretty striking. As I mentioned, we also look at other domains. So in terms of sleep and quality of life, the Pittsburgh Sleep Quality Index, a very commonly administered metric, looking at sleep quality. Here, lower numbers are better. Folks go from a 9.2 to a 5.8. It's 36% change. And when we look at the short form health survey, or the SF-36, the basic measure of quality of life, we see some rather compelling and striking significant improvements in areas like physical function, energy, emotional well-being, social function, and pain. This is interesting. Again, baseline to 4 weeks of treatment. These are pretty striking differences over time. The only thing we didn't see, and that's pretty interesting actually, because we think of them potentially as control areas, role limitations with regard to the physical role that someone has to perform, and general health. So in terms of some of the other areas where we might expect to see some change, we do. And in those two, we don't between baseline and 4 weeks. When we turn to cognitive performance, this is very similar actually to data that we've been collecting for some time in our longitudinal observational study that's been going for several years. We basically look at people who are interested in using medical cannabis for a range of medical or psychological or psychiatric symptoms or conditions. We look at them before they begin using cannabis, and again, over time, we go all the way out now for several years, but we published a paper looking at cognitive change over the course of 1 year. So we look at baseline to 3 months, 6, and 12. And what we found was basically some improvement over time. Again, those folks are choosing their own regimen. In this case, this is a clinical trial, and what we see is exactly the same. Patients are basically demonstrating stable or improved performance. They're not demonstrating decrements, especially in measures of frontal or executive function. This is just an example of some of the measures. The trail-making test part B, not significantly different. They're certainly not getting worse. For Stroop interference time, or time to complete a task requiring active inhibition over an overlearned response tendency, we see a statistically significant improvement with regard to time required to get the job done. The Wisconsin card sort is considered a gold standard measure of frontal function. Total correct, what we see is a statistically significant improvement between baseline and 4 weeks. We also see the same thing in the number of categories achieved on the Wisconsin card sort. So they're getting better. They're not getting worse over time. That's important, especially when we consider what we know to be the case in recreational consumers. Very often we see decrements in areas of executive function. We have some other measures of executive function. The letter number sequencing task and a task of phonemic fluency. Again, we're seeing either stable or improved performance patterns, not decrements here. Improvements after 4 weeks of treatment. Folks are going from a 12.5 to a 13.4. That's a trend. That's pretty interesting. Again, alternate versions of tasks are used, so we're not really thinking this is a practice effect, especially after 4 weeks, but certainly something to watch over time. I think that the expectation, perhaps, when we introduce something that's cannabis or cannabinoid-based is, I'm sure it's not necessarily going to make people better. I hear that a lot, and that's just not what we're seeing in the case of this particular clinical trial. So to conclude the CBD open-label clinical trial data, so far findings suggest that use of this custom-formulated whole plant-derived high CBD sublingual product results in less severe anxiety as measured by the OASIS and fewer anxiety-related symptoms as measured by the BAI and the HAM-A after 4 weeks of treatment relative to their own baseline. Self-reported improvements in other mood symptoms like sleep and quality of life were also noted, which may be related to reduced anxiety. It's funny, these things often coexist, so it's hard to disentangle them. I have often noted that sleep disturbance tends to happen in lots of different conditions. So it's not surprising that when we see sleep improvement, we also see other things improve and vice versa. Improvements on tasks of executive function were also noted. We certainly didn't see any worsening of task performance, which is really the important issue here, and no significant adverse events or intoxication were reported at all over the course of the trial. A definitive assessment of the impact of this novel treatment will certainly come from the double-blind placebo-controlled phase, which is, as I mentioned, currently underway, and we have this additional study arm that will now allow us to compare the full-spectrum product to a matched CBD isolate, which I'm very, very excited about. One thing that I mentioned sort of in passing is that we did monitor people for THC positive status over the course of the trial. And the reason for that is that many, many people are taking what they call, quote, CBD products. They say, oh, I'm just taking CBD. What they really mean is they're taking a product that's either high in CBD or marketed as such or contains CBD, primarily hemp-derived products. And the big question is, am I going to fail this random drug test since I'm taking CBD, which I think is a great question. There have been very few studies that have directly assessed whether the use of a full-spectrum high CBD product could yield positive results on a urinary drug test. And assessing cannabis use through the detection of THC metabolites. It's really, really important. So we monitored this status in this open label trial, and we used in-house or in-lab drug assays, and we then confirmed those data using GCMS quantification in pre versus post-treatment. We also looked at the relationships between THC positive status and demographic variables, as well as things like creatinine. Creatinine is typically reflective of kidney function and hydration. So after 4 weeks, remember our final sample was 14, not 15, folks. We had to get rid of one of them. Exactly 50% of our subjects, or our patients, 7 of them, were positive for THC metabolites after the 4 weeks. GCMS confirmed the assay findings, but indicated that the drug screen was actually more sensitive than its stated lower limit of detection. That's probably because of some cross-contamination from other cannabinoids. THC status was only significantly associated with creatinine. There was no association with any other demographic variable. For example, BMI. There was also no association between THC status and how much product people actually took. Remember, I mentioned that we do bottle weights out and bottle weights in, and the actual amount was somewhere around 35 mg of CBD that folks were getting every day, not 30. So you would think that people who had more might have had a higher chance of testing positive. That wasn't the case. So it's not amount of product, it's not BMI, it's not any other demographic variable. The only relationship we saw was with creatinine, a measure of hydration. Patients consistently using full-spectrum hemp-derived products then may test positive for cannabis use on urinary drug screens, which is something that people need to know. We definitely need studies with larger sample sizes to more thoroughly assess which variables, like how much product people are using, BMI, age, sex, race, other medication use, for example, how those things contribute to positive THC status in only some individuals, particularly those with higher creatinine levels. Importantly, and this is something I can't stress enough, this study product, if you look at the math, contained 0.02%. Let me say that again, 0.02% THC by weight. Remember that hemp-derived products can contain up to, that's right, 0.3% THC by weight. That's more than 10 times the amount of THC as in my current study product, and half of my sample was positive after 4 weeks of treatment. So it's often assumed that individuals who are using hemp-derived products will be negative for THC, but this may not be true, especially if assays are more sensitive than advertised, or they're being impacted by what I would call cross-contamination from other cannabinoids. Those assays are designed to look at the THC metabolites, but the presence of others may, in fact, create a little bit of a change. These findings certainly underscore the potential for adverse consequences. Loss of employment, for example, legal or treatment ramifications, despite the legality of hemp-derived products. They're legal, but that doesn't mean you won't test positive for THC, depending on what you're using. And if I had $1 for every person that said, but I'm only using CBD, and then they hold up this bottle, and it's not CBD, it's a broad spectrum product or a full spectrum product. I've also seen them turn positive with broad spectrum products. We have a number of other ongoing clinical trials at the MIND program, some of which are pending. We have INDs received for pain. We were just approved for anxiety in patients with glioblastoma and in patients with bipolar disorder, which is very exciting. We have some INDs pending for indications like sleep and so many more. What do I mean when I say many more? A number of different indications. So the MIND program will continue to support a range of studies that will generate ecologically valid, empirically sound data, examine the unique synergistic effects of cannabis and its constituents to determine the efficacy of cannabinoids for specific conditions and disease states, clarify the overall impact of cannabinoid treatment on physical and mental health, invest in studies designed to improve patients' overall well-being, and assess potential risk and harm, as well as patterns of misuse. It's not all about just the good. We also have to look at the potential, quote, bad or downside. Ultimately, we'd like to harness the therapeutic potential while minimizing risk and harm of cannabinoid-based treatment and exposure. We need to overcome barriers and conduct empirically sound research to draw clear conclusions. I always say, it's not one thing or the other. It's the good, the bad, and ultimately the truth. Without vision, we have no ability to move forward, so very often we're forced to think outside the box in order to move the field forward. And with that, I would thank you for your time and attention, acknowledge my extraordinary staff without whom I couldn't do any of this work. They are the most dedicated people ever. And I would, again, wish you a wonderful rest of your conference and be safe. Thank you again.